Hello everyone, I am delighted to welcome you to the annual Symposium of the Royal Society of Canada during its celebration of excellence and engagement. The University of Toronto is honored to be the presenting sponsor of this year's celebration. Let me take this opportunity to thank the Royal Society of Canada, U of T's Symposium Planning Committee, our speakers and panelists from universities and hospitals across the country, our partners in industry and the not-for-profit sector, and everyone joining us today. This year's symposium is focused on the legacy of insulin discovery, origins, access, and translation. It launches Insulin 100, a series of events to be held over the next 12 months to mark the centenary of one of the most important accomplishments in the history of medicine. The work of Banting, Best, MacLeod, and Connaught suddenly and profoundly transformed the lives of so many people around the world. It has saved countless more in the intervening years, and it reverberates today in the health of millions of people around the world who live with diabetes. But of course, there is still no cure for diabetes. It remains a leading cause of death, disease, and disability, and its prevalence is rising most rapidly in low and middle income countries. For all these reasons, the topic of this year's symposium is so fitting. We're celebrating an historic breakthrough, and we're redoubling our efforts to build on that breakthrough, to keep pushing the boundaries of research and innovation, to improve the quality of care provided to patients, and to address challenges of health equity at home and abroad. Clearly, there is still a great deal to be done to prevent and treat, and maybe one day to cure diabetes. But we have many reasons to hope. The story of insulin is a brilliant example of the power of collaboration, demonstrating how a university, its hospital partners, and a pharmaceutical company could work together and change the world. It's a story that continues today, as we see in the exciting work being done by the participants in today's symposium. Leading Canadian scientists, clinicians, and innovators, and the brilliant students who are following in their footsteps, all working towards a healthier and happier future. Thank you for your dedication to that great cause. I wish you all a stimulating, informative, and inspiring day.
Hello, it's my pleasure to welcome all of you to Toronto for this virtual gathering. When we first proposed to the Royal Society of Canada of having a meeting here in Toronto in order to celebrate the launch of Insulin 100, a celebration of the discovery of insulin and its impact worldwide, we could not have imagined what 2020 would actually be like. While it would have been wonderful to have everyone here, as with everything else this year, we have had to adapt. I would like to share with you some of the lessons from the story of the discovery and distribution of insulin that are relevant to the present day, and particularly relevant to the management of the current COVID-19 pandemic. Recall that the lives of those involved in the discovery of insulin were shaped by the 1918 pandemic. I will briefly recap a few highlights and draw the connections to the present day. The story of the discovery of insulin is the story of science and knowledge transfer as we now know it. It was not a simple eureka moment. There are legends of Banting waking up from a dream and furiously writing down, ligate pancreatic ducts of the dog. Keep dogs alive till acini degenerate, leaving islets. Try to isolate internal secretion of these and relieve glycosuria. That notation in his diary is certainly very real. The story of the dream, maybe not. Certainly, Banting had been informed by the work of many before him. And as came to be, following the awarding of the Nobel Prize to Banting and Best, there was a great deal of controversy about others who might have shared in the prize who had been working on similar concepts. Science is a result of collaboration. It's the result of people building on the shoulders of giants. The story of the discovery of insulin is also one of long and tedious work. It's one of taking risks. It required perseverance. It entailed conflict, even fistfights, resulting in bloody noses between the scientists that were involved. And what story of inter-university collaboration would not be complete without patent disputes? They confronted failure on many occasions, but they found ways to get around the problems that they encountered. They brought in people from many different disciplines. No one researcher could have done what was accomplished alone. There was collaboration with the private sector, with governments, with foundations. It required an all of society response, just like we're seeing with COVID-19. In order to get insulin out once it had been developed, global collaboration and networks had to be built. And in an era before there was regulatory affairs at Health Canada or the Food and Drug Administration in the United States, they had to manage the quality assurance process and ensure that the insulin that was being produced was safe and effective for those that were receiving it. Imagine the questions that we have now around the production of a vaccine. These issues that they grappled with 100 years ago continue to dominate our discourse today. Turning briefly to the story of the discovery of insulin, we often think of Frederick Banting and Charles Best. Less well known is the work of John MacLeod and James Colloque, who also shared in the work and ultimately shared in the winnings of the Nobel Prize. And there are many more that I do not have the time to go over who were involved. The clinicians, the people working in the labs, the people working in the pharmaceutical companies that help sort out how to produce insulin at scale. But we'll focus on the four key individuals. Banting was a physician in practice in London, Ontario. As I mentioned, he had these ideas and he managed to visit with MacLeod, who was chair of physiology at the University of Toronto. On November 6th, 1920, just about 100 years ago, he presented his idea on isolating a treatment for diabetes from the pancreas. MacLeod took a risk and gave him a small lab, equipment, and experimental animals. Banting did not have to write a grant and wait months and maybe repeat the process several times before getting this access. He was also provided with a medical student to assist him. Best and a colleague named Noble flipped a coin to see who would work with Banting first. Noble lost out on the Nobel Prize through a coin toss. Through the spring and summer of 1921, they worked to prepare a pancreatic extract that they showed could reduce sugar in deep pancreatized dogs. I'll note there were no animal care committees. On July 30th, 1921, they were able to successfully isolate a serum that they injected into the dog and showed that the blood sugar was reduced by 40% in 1 hour. Unfortunately, they couldn't repeat the experiment in the weeks that followed. With the advice of MacLeod, they collaborated with Connaught, who was a biochemist on sabbatical from the University of Alberta, who helped to purify the extract to be used on dogs successfully and then on patients. Their first test in a human was at the Toronto General Hospital on January 11th, 1922. Of note, there was also no ethics review board at the time. Banting and Best did not originally want to patent insulin. There was a prevailing view that medical researchers should not profit from their discoveries. They should make them available broadly, what we would think now as an open science approach. However, there was concern about ensuring access broadly at an affordable price and ensuring quality. If they didn't file a patent, many different companies could have started to develop and manufacture insulin. There were also disputes amongst the inventors about what to do. The University of Toronto people were worried that Connaught would file a patent through the University of Alberta. So, frictions between institutions live on. John Fitzgerald, who's the head of the Connaught Labs at the University of Toronto, facilitated a very important agreement amongst the four inventors. They agreed not to take out any patents on their own or assign them to a commercial pharmaceutical firm during an initial period of working with Connaught. Second, they agreed that they wouldn't change any of these provisions without first discussing amongst Fitzgerald and the four collaborators. As the inventors did not have the capacity to manage this themselves, they assigned the patent to the University of Toronto for $1. And then the university had its Connaught laboratories, which were part of the institution, lead negotiations with manufacturers. Eli Lilly offered to help in ramping up manufacturing, as Connaught did not have the scale to meet the demand for this vital drug. The university, seeing that this was far beyond the usual work of the institution, set up a special insulin committee of the Board of Governors. Its role was to ensure that at the earliest possible date, adequate supplies of potent and non-toxic preparations of insulin will be constantly and readily available at reasonable prices all over the world. They created a very innovative patent pool to share in the advances. Everyone that was given rights to the license to use insulin had to assign any of the patents that they took out on processes around manufacturing back to the university, so that all of these patents could be pooled and shared with everyone globally. They found partners around the world, the Medical Research Council to distribute it in the United Kingdom, and they worked with Hans Hagedorn to assign rights for Europe, which led to the creation of what is now known as Novo Nordisk in Denmark. And Connaught Labs was able to scale up production, so it was able to supply many parts of the then British Empire, as well as the rest of the Americas outside of the United States, which was supplied by Eli Lilly. All the royalties that came in from this went into the Connaught Labs and supported research and quality assurance work. They had a series of presentations. The first one was at the American Physiological Society in December 1921. They published their preliminary report in the Canadian Medical Association Journal in March of 1922. And then in May of 1922, they delivered a series of lectures at the annual meeting of the Royal Society of Canada. So it's a privilege to be here to celebrate once again with the Royal Society of Canada. The Nobel Prize was awarded in 1923 to Banting and MacLeod. Banting was furious but Best was not named. So he offered to share his half with Best. MacLeod responded by sharing his half with Connaught. Relations were not good between the four men, but they can see how they all acknowledged the contributions of each other. At the gala dinner that was held in Toronto later that fall, it was stated, "In insulin, there is glory enough for all." 100 years later, there are millions of people around the world that continue to benefit from this discovery. The technology evolved, and once the Toronto patents expired, the ability to control price and accessibility was lost. Insulin prices remain a significant issue today. As a result of globalization, the university could not compete as a global biopharmaceutical company, and the Connaught labs were sold. The legacy for the university is our very generous Connaught Fund. But the legacy for Canada is less rich. In this era of COVID-19, we see that we need to have our own manufacturing capacity for biopharmaceuticals, and there are now efforts to rebuild it. There are many other important lessons from the story of insulin, particularly around the importance of being open in science and the importance of collaboration across disciplines. These are all things that the Royal Society of Canada celebrates, and it's a pleasure to have shared these thoughts with you today. Thank you.
Hello. Thank you very much, Professor Goel. Welcome everybody to the first session here, introductory session of this webinar. My name is Christopher Ruddy. I'm a medical historian, kind of in private practice. I have an adjunct appointment at the University of Toronto Dal Lana School of Public Health. And I did my PhD with Michael Bliss. So I kind of channel my Michael Bliss these days. I've been involved with a number of projects on the history of insulin, curating various exhibits, including the one at Mars you might have seen. And I'm currently the lead historian on the Defining Moments Canada Insulin 100 project, of which the U of T is a major partner, along with Sylvie Pasteur and Health Heritage Canada. And the second session of this webinar is through Defining Moments Canada. So I'm here to introduce this first session of the webinar, which is called Insulin Discovery and the Intersection of Academia and Industry. And we have two speakers who have pre-prepared videos. We have Gary Lewis, and we also have Elizabeth Neswald. And I'll introduce each separately. We'll first do Gary. I can welcome both of you before we get into that. Hello, Gary and Elizabeth. Welcome to and for contributing to this session. I'll start with Gary. His paper will be called Discovery of Insulin Revisited, Lessons for the Modern Era. And Gary, Dr. Lewis did his training, basic medical training in South Africa, and was followed by specialty training at the University of Chicago in internal medicine. Oh, sorry. Excuse my phone in the background there.
Thank you very much for your introduction, and thanks for the kind invitation to present to you a revisit of the momentous discovery of insulin in Toronto in 1921, 1922. And I'm particularly interested and will share with you some of the lessons from that discovery that are still relevant for the modern era. I would like to acknowledge the work done by two historians, the late Professor Michael Bliss and Chris Ruddy. I have drawn quite heavily on their work, and I really wanted to thank them for that. So this was diabetes before insulin. It was shocking. It was universally fatal, and tragically it often affected children, mostly affected children, adolescents, and young adults. And these terrible pictures look like concentration camp survivors. They would waste away a loss of both body fat and muscle tissue and develop something called ketoacidosis. There was really no treatment. Many diets were tried. many kinds of treatments. Nothing was successful before the discovery of insulin. This is a wonderful quote from Michael Bliss from his famous book, The Discovery of Insulin, and I'll read it to you. The discovery of insulin at the University of Toronto in 1921-1922 was one of the most dramatic events in the history of the treatment of disease. Insulin's impact was so sensational because of the incredible effect it had on diabetic patients. Those who watched the first starved, sometimes comatose patients with diabetes receive insulin and return to life saw one of the genuine miracles of modern medicine. They were present at the closest approach to the resurrection of the body that our secular society can achieve, and at the discovery of what has become the elixir of life for millions of human beings around the world. So discoveries rarely come out of the blue, of course, and what was the body of knowledge localizing insulin to the pancreas prior to 1921? Well, Paul Langerhans in 1869 was studying the pathology of the pancreas, and he discovered these clusters of cells in the pancreas, subsequently became known as the islets of Langerhans, and these were floating in the pancreas. So the pancreas is this organ that sits behind the stomach and in front of the spine, and most of the pancreas actually consists of these acini. And acini are glands that make enzymes, and the enzymes travel through these little ducts, which then coalesce to become a single large pancreatic duct, and then takes the enzymes to the first part of the intestine called the duodenum. And these enzymes are responsible for food digestion. So the pancreas consists of sort of these two parts, the acini, which make enzymes for food digestion, and then Paul Langerhans did not know the function of these islets, and these became the focus of activity over the next few years. But right back to 1889 and 1893, investigators started to hone in on this pancreas and did experiments in dogs. And they reproduced the diabetic state by removing the pancreas from dogs. And they hypothesized that this was due to loss of an internal secretion of the pancreas rather than the pancreatic exocrine secretion. Actually, they performed many experiments, some of which were partially successful in reducing the glucose, and they measured the glucose in the urine. So they made the dog diabetic by removing their pancreas. And when the blood sugar rises, it spills over into the urine and can be detected there. And so they were measuring, and then they were making an extract of the pancreas and injecting it back into the dogs and looking at how the dog did. And they had partial success. In fact, over the next 2 decades, even a number of experiments were done in humans, injecting this extract into humans, but the human experiments totally without success. Lionel Opie, working at Johns Hopkins in 1901, was looking at pathology specimens, looking at the pancreas from individuals who had diabetes and died of diabetes. And he noticed inflammation in the islets of Langerhans in these individuals. So he drew this pathological connection between diabetes and damage to the islets of Langerhans. And many investigators worked on this in the next 2 to 3 decades. I mentioned Minkowski and Mehring, Hayden, Rennie and Fraser, Zulsa, Scott, Kleiner and Meltzer, and Paulescu from Romania. And in fact, they had variable success. Paulescu published his results right at the time that Banting and Best were starting their experiments, and he had success in lowering the glucose in dogs. In fact, there was a controversy that went on for many, many years after the awarding of the Nobel Prize in 1923, when many felt that Paulescu was overlooked and should also have shared the Nobel Prize with the Toronto group. And we'll talk about that a little bit more. But certainly, the work of the Toronto group was did not come out of the blue and really was based on a large body of work isolating an important internal secretion to the pancreas. So Banting was a struggling surgeon working in a town called London, Ontario. It's 200 kilometers west of Toronto. He was really struggling to set up a clinical practice, and he was giving some lectures, and he actually read some interesting articles. And the story goes that he woke up at 2 o'clock in the morning and jotted down this idea from his readings. And you can see the original memo that he wrote. And what it says is, ligate the pancreatic duct of dogs. So ligate means to sever or tie off or to cut. Keep dogs alive until the acini. Remember, the acini are the part that secrete the digestive enzymes. So wait for those to degenerate, leaving just the islets of Langerhans. And after that, try to isolate the internal secretion from the islets so that it would not be destroyed by these pancreatic enzymes. So this was Banting's idea, or one could call it his hypothesis. And we'll talk about a hypothesis in a little while. It turns out that was not correct. And in fact, there were a number of misconceptions or errors in this. But most important of all is that the pancreatic duct does not have to be ligated in order to isolate this internal secretion. So Banting spoke about this to some of his colleagues and was encouraged to go to Toronto, the University of Toronto, to speak to a famous carbohydrate researcher, J.J.R. McLeod. I'll show you his picture in the next slide. But the University of Toronto was actually a fantastic place to do experiments of this type. Not only was McLeod there with very established labs and know-how in the area of carbohydrate metabolism, but in fact the medical school building housed a very established and research-oriented medical faculty. The Toronto General Hospital, where I currently work, was across the street, and that is where the first injection of insulin was given to a human. The Hospital for Sick Children across the street from that, where many of these children were admitted to hospital with type 1 diabetes. And very importantly, the Connaught antitoxin lab, which was actually created in 1914 during the war to create diphtheria antitoxin. And the Connaught antitoxin lab was in the basement of the medical school. It became very, very important in the early production of insulin. So this is J.J.R. McLeod. He was a native of Scotland, and he was the head of the Department of Physiology, and he had a lot of resources at hand. And Banting, the surgeon from London, Ontario, came to speak to him. He was very skeptical initially of Banting's idea, but he did relent, and he gave Banting resources to start his experiments in 1921. So this is the lab. He gave him dogs to do the experiments on. He also gave him the assistance of an undergraduate, a graduate student, Charlie Best, to work with Banting. This antiquated equipment to measure blood glucose is actually very important, because although it is very dated, obviously, it actually gave these experimenters a huge leg up on those who were previously doing experiments. So being able to measure glucose in small aliquots of blood and repeated samples much more precisely than any of their predecessors could do. They started the experiments in May 1921, and lo and behold, by August that same year, they started to have this success. So here you can see the chart of the blood glucose of a dog that was administered this. So what they did was they ligated the pancreatic ducts. They waited 4 to 7 weeks. They took the pancreas out. They mashed up the pancreas. They purified and tried to isolate this internal secretion. They gave it back to a dog that had had its pancreas removed and was very sick and diabetic. And you can see this reduction in the blood glucose of the dog, and Banting's lab notes as well. So success by very early I can tell you as a researcher, and many of you listening to this have done your own research, and this certainly never happened to me. It takes years often to get results, and not quite as stunning as these results. So late in 1921, there was a biochemist, very, very skilled biochemist by the name of Connaught, and he was on sabbatical from the University of Alberta, on sabbatical in McLeod's lab, and he had some experience in purifying secretions, and he actually got to work. So McLeod asked him to become involved in this project in about November or December of 1921, and he used an alcohol extraction method, and it's like baking a cake. It's got to be just right. So making the exact percentage of alcohol, approximately 90%, to extract the impurities and leave the active substance in solution, and change the pH this way and that way. He started to inject this extract into rabbits, which was much, much quicker and less expensive than doing it into dogs. And they felt they had a good extract by early 1922. So Leonard Thompson, a young boy, 14 years old, didn't look like this at the time, was very, very sick. He had ketoacidosis, he was wasted, just like the pictures I showed you. And on the 11th of January, they gave him an extract, very large volumes into each buttock, very painful, actually developed sterile abscesses at the site of injection, and had no effect. So they went back to Connaught and they said, you got to work harder, you got to clean up this extract and give us a more purified solution, which he did. And on January the 23rd, gave this new formulation to Leonard Thompson with excellent results, in this case, looking at reductions in glucose in the urine. So that was the first successful administration of this pancreatic extract to a human. Leonard Thompson started to perk up. He felt better. He was less sick, and over time received these insulin injections, gained weight, and looked like that later on. The press got caught whiff of what was happening, and were very excited. Here is a headline from the Toronto Daily Star, now called The Star. Toronto doctors on track of diabetes cure. Diabetes work epoch-making, save physicians. So we've drawn this timeline of exactly where in the discovery of insulin did the work of this Toronto group, Banting, Best, McLeod, and Connaught. Where did it fit in? And this was in 1921 and early 1922. Well, I've already told you about the work of Paul Langerhans, discovering what became known as the islets of Langerhans, and then the work of, I won't name them all again, but all of these outstanding investigators who honed in on the pancreas with greater and greater success leading up to the work in Toronto. But in actual fact, this was not really the discovery of insulin. It was the purification of insulin to a level that it could be safely administered to humans and cure their diabetes. But the discovery involves more than that. It involves discovering the amino acid sequence of the protein, of the peptide, insulin, which came later, the genetic code of the insulin gene, how insulin is secreted from the cell. And even this, and these were Nobel Prizes that were subsequently awarded over the century following this purification of insulin. I'll just mention Rosalind Diallo working on the radioimmunoassay, which was a very important discovery in the 1960s, allowed investigators to then measure these minute quantities of insulin in the blood and begin to really understand its secretion and its action. So many other discoveries following the work of the Toronto Four, which were critical in the actual discovery of insulin. So the question comes up, what was the 1921-1922 work in Toronto the true discovery of insulin? And I've told you that not really, it was actually the purification, but what is well beyond dispute is that Banting, Best, Collop, and MacLeod were the first to develop an insulin preparation as an effective therapy for humans affected by diabetes, and no other investigators or groups can lay claim to that accomplishment. So what were the Toronto researchers' success factors compared to their predecessors who made great advances over the previous 3 decades? Well, firstly, as I've indicated to you, there was a large body of knowledge that had already localized an internal secretion to the pancreas. Secondly, Banting was not simply a curious researcher. In fact, you could hardly even call him a researcher, but he had no training in that field, but he wanted to, if possible, isolate a form of internal secretion that will be of use in treating diabetes. His drive, sheer determination, perseverance, and a laser-like focus on developing a successful treatment for diabetes was really the success factor. I already mentioned there were advances in determination of blood glucose. They could measure it much more precisely. In fact, it's entirely possible that their predecessors had more success than they thought they had, or they knew they had, if they could have measured glucose more successfully, more precisely. Banting and Best worked with a very experienced carbohydrate researcher, MacLeod, in a well-resourced research university. And the experienced and brilliant biochemist Collop optimized the alcohol extraction method to isolate insulin relatively free of toxic impurities. So really all 4 of them played a very important role and contributed to the success. And as I'll mention, once they had discovered insulin, there was now a huge need. People were dying, children were dying, and everybody needed insulin. And this is where the collaboration with industry was very important to scale up production. So I'm quite interested in this. It's a very common pitfall. We see it all the time. Okay, I've probably been guilty of this on more than one occasion. And it is the believing one's hypothesis to the point that it impairs judgment. So we come up with an idea. We think it's a brilliant idea. And it's very important in research that it's not the idea. You can think of any idea you want, but it's the testing of that idea. So I can tell you, interacting with trainees, and many of you may have had the same experience, often you'll hear a trainee say, we set out to prove the hypothesis. And you have to stop them and say, no, that is actually not what you were doing. You were not trying to prove the hypothesis. You were testing the hypothesis. And in actual fact, more technically, you're testing the null hypothesis. So the hypothesis or the idea may be right or it may be wrong. It doesn't really matter. Everything you do is going to be an advance to science. But this was a classic case of Banting believing his hypothesis or his idea to the point that it actually blinded him. So what really happened was towards the latter part of 1921, they had lots of delays in generating duct-ligated dogs. It took 47 weeks, and they were running out of extract. And they needed to do these, what's called longevity experiments in dogs. Dogs were made diabetic, and what MacLeod wanted them to do was not just lower the blood glucose once with a single injection, but administer this extract, which came to be known as insulin, on a repeated basis to keep the dogs alive. And they were running out of the extract because it took too long. So they began to use the pancreas from only partially degenerated pancreas, and they actually had some good results. But however, this is the point that I wanted to make here, is at first they actually failed to recognize their own findings. And in fact, I'll just go back a little bit. They failed to recognize, and so in a subsequent publication, they claimed that the duct-ligated pancreas worked better, more effectively than that was not degenerated. But that was not true. It was not indicated in their findings. And Banting read a paper towards the end of 1921 showing that newborn or fetal pancreas had more plentiful islets. They actually obtained fetal pancreas from a local abattoir with very good success. So they started to pivot away from doing what they thought they had to do, which is ligate the pancreatic duct. And finally, they resorted to using a pancreas of non-ligated adult pancreas from a dog with a very successful decline in blood glucose. So now the race was on to supply insulin to the world, and as you can imagine, the stress. And this is where the Connaught labs came in, and it's very primitive insulin production in the early days in the basement of the medical school building. This is an old insulin vacuum still. And to the rescue came industry. Okay, so Eli Lilly in Indianapolis and Klaus, who was from Eli Lilly, caught wind of the Toronto experiments earlier in 1921 and was interested in it and was following it. And so he made contact with the Toronto researchers, and they came to an agreement that Lilly would assist and would produce insulin and would make it available for all of those who needed it in the first year at simply at cost. And in actual fact, huge advances were made in the manufacture of insulin because there were failures at the Connaught lab in 1922. They could not supply the world with insulin. And there were, for example, the isoelectric focusing was a huge advance, and the purification of insulin. And by 1923, Lilly was able to manufacture and supply at least all of North America. And the Toronto group also licensed the insulin to the Royal Society in the UK and to the beginnings of what has now become a global leader, the Novo Nordisk, the early Novo Nordisk group in Denmark as well. There was spreading excitement encouraged by Leonard Thompson's progress, and clinical trials were beginning in select places, in Toronto, in Boston, in a few places. And you had these resurrections, these wonderful success stories of a little girl who looked like that, and then after months of treatment looked like that, much, much better, healthier. And Banting really became the superstar. New York excited over new cure for diabetes, gives Banting all the credit, etc. And these wonderful stories to the doctor who cures diabetes from a young girl who was saved by insulin. Some of the early patients were famous. So Elizabeth Hughes was the daughter of the Secretary of State of the United States, wonderful young girl who was one of the early patients, and many of the others here. And making insulin at a lower price was very important. The price actually declined from 1922 to 1942, and we'll talk a little bit about what happened subsequent to that. This is the staff of the Connaught lab, and this is one of the early insulin vials from the Connaught labs. Why did the discoverers of insulin, this is well known, they chose not to profit personally from the discovery. What was that all about? So the discoverers sold their rights to the University of Toronto for $1 a piece, and the university filed an application for a patent. They felt that providing their patent to the university would prevent their discovery from being commercially exploited by a single company. And justification for filing a patent was that when the details of the method of preparation were published, anyone would be free to prepare the extract, but no one would secure a profitable monopoly. And the discoverers were very reluctant to be associated with patenting on ethical grounds that commercialization might make their discovery unavailable on a widespread basis. Banting famously declared that insulin does not belong to me, it belongs to the world. They had very high ethical concern that ensured that even the most financially destitute of patients would not be deprived of insulin by commercial forces. And the discovery of insulin provided a formative test case for the relationship between academia and industry. And unlike today, in which commercialization of medical discoveries is a point of pride for researchers in universities, in 1921-22, researchers in universities felt uneasy about profiting from medical discoveries which were first and foremost to benefit humanity. So 100 years after its discovery, insulin is still unavailable, unfortunately, to millions of people in need. Globally, 1 in 2 people needing insulin lack access. Improving insulin availability and affordability is complicated. It needs to be addressed through national and global actions, including prioritizing the supply of more affordable human insulin, increasing competition through the use of lower-priced, quality-assured biosimilars, negotiating lower prices from manufacturers, and improving distribution systems. The WHO Global Report on Diabetes in 2016 states that people with diabetes who depend on life-saving insulin pay the ultimate price when access to affordable insulin is lacking. This was one of the fastest Nobel Prize awardings. It was awarded in 1923, just over a year, about a year and a half after the first successful human administration. And it was interesting that the names Banting and Best, of course, are most associated with the discovery of insulin, but in fact, the Nobel Prize was awarded to Banting and MacLeod. Now, by then, there was some acrimony between Banting and MacLeod, and Banting immediately split his award with Charlie Best, and MacLeod split it with Connaught, and in the end, with all 4 of them benefiting from this award, it was probably the correct attribution because all 4 played a very important role. And finally, just to say that it's not as if nothing's happened over the last 100 years. So there's been tremendous advances with purification and concentration of insulins, modified insulins that have delayed absorption. This makes it very helpful with the treating of diabetes. Advances in glucose self-monitoring, which you're going to hear about as well. Very, very interesting, big advances over the next century. Discovery of other ways to monitor how the person with diabetes is going, something called the hemoglobin A1C, which measures the last 3-month average glucose in the blood. Improved insulin delivery advices, pens and infusion pumps. Manufacture of the first human insulin in the 1980s, a major advance. And ultra-rapid and ultra-long-acting insulins, as well as continuous glucose monitoring. However, there are still many challenges for the person who needs to take insulin for the rest of their life. And the Holy Grail will be, we're very, very close to this, almost there. Actually, it even exists, which is what's called a closed-loop device, a pump that infuses the insulin under the skin, and it can be programmed to infuse just the right amount, and continuous glucose monitoring device, which was a big engineering hurdle that has been overcome, and that can give continuous glucose readings. And then the connection of the two by an algorithm, a mathematical algorithm that can say, here's the sugar, here's how much insulin to deliver, a sort of a plug-and-play artificial pancreas. And I can tell you that the delays because it takes billions of dollars to have these devices approved by regulatory authorities. And many people with diabetes are not waiting for that. They've actually jerry-rigged their own a closed loop. We call this a closed loop system. They're not approved, so we can't endorse this. But many people already with diabetes are using closed loop systems. And then, of course, cell-based therapy. So if we can take a stem cell and make it into a cell that makes insulin like these ones and transplant them into the body, that can be almost as good as a pancreas that's not diseased. And finally, thank you very much for your attention. This presentation was based on a paper that I wrote with my colleague Patricia Brubaker, professor in the Department of Physiology at the University of Toronto, and the publication date for this in the Journal of Clinical Investigation is January 4th, 2021. And thank you very much for your attention.
Thank you very much, Dr. Lewis. Great presentation. And I want to, sorry I got interrupted in my introduction of you earlier on. My phone rang. I couldn't control that. But I'll get back to that and round up your introduction when we get to the question period after Elizabeth's talk. So I just want to move on to Elizabeth's talk right now. So Elizabeth Neswald, her paper is going to be called Shifting Expertise, Shifting Responsibility, Developing Glucose Monitoring Systems After the Discovery of Insulin. And Elizabeth is an associate professor for the history of science and technology at Brock University in St. Catharines. She's been a scholar in residence and fellow at the Max Planck Institute for the History of Science, and a visiting scholar in the History of Science Department at Harvard University. And most recently, she's the Doan Fellow at the Science History Institute in Philadelphia, where much of the research for this talk was done. And she's also a chair of the Forum for the History of Health, Medicine, and Life Sciences and History of Science Society, and has numerous publications in the history of science and medicine. Her research in the past few years has focused on the early history of nutritional physiology and metabolism studies and history of diabetes management in the 20th century told through objects and artifacts. So thank you very much, Elizabeth, for joining us, and on to your presentation.
Thank you so much to the organizers for inviting me to speak here at the celebration of the discovery of insulin, and to everyone in the audience for coming to listen. I also thank my co-panelist for his wonderful presentation on the history of insulin from its discovery to the present day. This means that I can focus on other aspects of insulin history. Insulin was a game changer in the treatment of diabetes, but it could not do this alone. As medical publications and manuals for diabetics and physicians in the 1920s emphasized, insulin was dangerous. Administered into low a dose, it was ineffective, but into high a dose, it was potentially a killer, leading to diabetic coma. In order to be an effective therapy, the insulin dose needed to be carefully matched to the individual diabetic's needs for close monitoring and constant adjustments. This is what I want to talk about, the history of urine sugar and blood glucose monitoring in the 20th century. And as you'll see, it is a colorful history because for the first several decades, it was based on visual analysis using reagents and indicators. It was also a history of shifting expertise and shifting responsibilities. It has often been noted that responsibility for urine and blood sugar monitoring moved from physicians to patients over the course of the century, but the story is not that simple. I look at it more as a redistribution of expertise, with different groups contributing their skills to the innovation of monitoring systems. That diabetes and sugar was somehow connected was recognized several millennia before the discovery of insulin. Ancient physicians tested for the presence of sugar in urine by seeing whether it attracted ants, or they added yeast to see whether it fermented. By the late 19th century, the amount of sugar could be roughly quantified with a simple saccharometer, like this one here. Fermentation generated carbon dioxide, the gas displaced the liquid, and the amount of displacement could be read off a scale and gave a rough estimate of sugar content. The saccharometers were used by physicians, especially in small rural practices, into the 20th century. From the early decades of organic chemistry in the mid-19th century onward, chemists specializing in medicine and physiology began developing reagent-based methods of analysis. The first relatively reliable one, Benedict's solution, was introduced in 1911, and it was based on the way that copper sulfate and sugar reacted in a heated solution. Important was that this was a fairly simple test. Urine was added to the heated solution, and if sugar was present, it formed a precipitate. The solution also changed color, but that was not considered important. What was important was that the reagent was stable enough that it could be bottled and sold. And this meant that patients could do their own urine tests to monitor their diabetes. Elliot Jocelyn, who was the leading American diabetes specialist in the early 20th century, even claimed in 1916 to have taught the test method to a child. At this time, the only treatment for diabetes was strict dietary control, as the photographs of children in the previous talk showed. And even that could only prolong life for usually a few months. Urine sugar testing was done for diagnostic purposes and at intervals to check whether the diet needed to be adjusted. The discovery of insulin and the development of insulin therapy radically increased the need for regular, accurate, quick, and simple sugar tests. So prior to insulin, diabetics had often spent long periods in specialist clinics and hospitals, but insulin moved everyday diabetic care to the home environment and a general practitioner's office. Because Benedict's solution was stable, it could be mass-produced and marketed to these physicians in test kits, like this Eli Lilly test kit number 1. So marketed in the 1930s, it was sold to physicians to do diagnostic tests, and it became the standard kit for small medical practices. So the cover image illustrates the process. As you see, all necessary chemicals were pre-prepared, the chemical apparatus is supplied, and there's a set of instructions on the inside cover to explain how to use it. This kit did 2 different kinds of tests with one solution. So it did the usual qualitative test, which just assessed for the presence of sugar through precipitate, and it also did a quantitative test, which measured how much urine needed to be added to the solution to turn it from blue to clear. And this was tricky because it depended on lighting and skill and was also very time consuming. So the analysis methods I have described up to now were designed for medical professionals, primarily for diagnosis and occasional monitoring. Soon after the introduction of insulin, it became clear that it was not enough to test urine weekly or even once daily. Ideally, it would be tested multiple times a day. So making the frequent tests manageable required changes to the way that they were done, and these changes redistributed skills and responsibility for testing to various actors. And this is where the next chapter begins, the development of testing modes that integrated the chemical knowledge of reagents and reactions so that the test could be done by a wider range of users. So already in 1929, chemist Abraham Scheftel was proposing a faster and easier method of analysis. So he recognized that if the precipitate in the qualitative Benedict's test was removed, the color changes themselves could be used to measure the quantity of sugar in the urine. So as he described it in his patent, a given amount of glucose in the solution will always correspond to a given shade of green. So color could provide the scale, and it would be possible to make a standard color chart that graded colors to the corresponding percentages of sugar in the urine. So this shift from measurement through titration to measurement through colorimetric methods fit in well with contemporary trends in biochemistry through the increasing use of color indicators. I'll talk a bit more about that later. So around 1940, the Eli Lilly company produced the sugar test kit number 6, which was based on Scheftel's method. The Eli Lilly chemists had integrated all of their skills for measuring and heating reagents into tablet form. So all the user needed to do was to mix urine and water in a test tube, drop in the reagent tablet, light the methanamine tablet, let the mixture cool, and then interpret the color of the results according to the color scale that came with the kit. And this was a small kit in a carrying case that was easily portable. Although it was still designed for medical professionals, it was quick and easy to use and suitable for home analysis. And what this illustrates compared to the earlier methods is that the user no longer needed to perform the chemical operations. They just needed to learn to accurately compare colors. And this is something that needed its own kind of training. So these changes were happening in the decades when the major pharmaceutical companies were starting to emerge from the wild field of quack cures and dubious supplements. And that had been Eli Lilly's beginnings as well. So Miles Laboratory had similarly dubious beginnings, but then they had their hit with Alka-Seltzer, which brought the company a brand name. So in the 1940s, they decided to enter the urine sugar analysis field too. Walter Compton and James Trenier, two of their chemists, took the techniques and the low humidity lab that they had developed for Alka-Seltzer and applied them to the development of effervescent reagent tablets for urine sugar analysis. Their new diagnostic test, Clinitest, was introduced in 1941. So Clinitest was one of the first tests made specifically for diagnostics. Although developed primarily for physicians, it quickly became clear that this kit was also suitable for diabetic patients. It went a step further than the Scheftel test in integrating chemical expertise into the test materials themselves. All the user needed to do was combine urine and tablet and then compare the results on the color chart. So this was the kind of test that could be done easy, quickly, and frequently at home. So all the methods I've discussed until now have been based on modifications of Benedict's copper-based solution. In 1956, Helen and Alfred Free at Ames Labs developed a new reagent based on glucose oxidase. What was unique about this new indicator was that it was mixed with gelatin and it could remain stable no matter what form it was in. So it remained stable in the form of a solution, tablet, powder, or dried onto other substrates. So Helen Free describes testing how it reacted with glucose when it was dried on such diverse materials as aluminum foil, pipe cleaners, paper clips, polystyrene, polyethylene, wood, paper, plastic, and rayon. She and her husband decided to use thick paper as the substrate and then cut it into small strips. So in an interview with the Chemical Heritage Foundation, she describes the early production process and the improvised setting. And this is worth quoting in her words. So I quote Helen Free. We used to have a card about half the size of an 8.5 by 11 inch paper, and we'd cut it with scissors and dip it into the solution and then let it dry. We'd have these cards put in a rack that had slats on 2 metal rods through the holes in the slats. We would put them in here, slap another slide up against it, put another reagent card in there, slap another slide against it, and then tighten those claps. And then we had this rack of cards that would stick up. And then we would dip that into a big pan of reagent and send it through the dryer. So these strips came onto the market as clinistics in 1956. They were the first dip and read urine tests. A similar type of test, the test tape, was manufactured by Eli Lilly and came out around the same time. Now, the main consideration behind these methods was the convenience and ease of use. And this is something that Helen Free also noted. So older methods needed some kind of skill and familiarity with the use of measuring equipment and the use of liquid reagents, which were potentially toxic. Dip and read test strips mark the end point of a particular development. So all of the chemical expertise that users needed in the early tests had now shifted to the chemists in the laboratories. So the user was presented with this knowledge materialized on a tiny strip of paper. And they did not need to do anything except dip a strip in their urine and compare the color of the strip with the package to compare the results. And this basic technique of dip and read strips has not changed significantly since the 1950s. So up to now, this has been a history of urine sugar testing. The history of blood glucose testing runs parallel, and they meet in the 1960s in the dextrosticks test strip. But I want to look at a different area where they traveled similar tracks first, and that's the question of standardization. Because for these testing methods to work without relying on significant expertise and experience, there had to be an easy and reliable way to interpret the results. So I mentioned earlier that much of the development of urine sugar tests depended on the use of indicators in biochemistry. So I want to go back for a moment to Otto Folin, who is often considered to be the father of biochemistry. So Folin became acquainted with an apparatus called the colorimeter during a visit to a brewery in 1897 when he was studying in Berlin. This apparatus had been designed by Louis-Jules Dubosc in the 1850s, and by the 1880s, it was in widespread use for the assessment of dyes, fuel, and liquor, among other commercial products. So one side of the colorimeter has a substance that's being analyzed, and the other has a standard sample. And the user looks through the eyepiece and can see both together and calculates the difference in concentration through a scale on the side of the apparatus. So Folin brought this instrument into the clinical chemistry lab, and he developed special versions to analyze samples, so biological samples. Now, the connection between Folin and colorimetry is so strong that the model that is on the slide, the BNL colorimeter model, one that he helped design, is also in the background of his official portrait. And you may also have noticed one in the slides in the previous talk, because Banting and Best used this kind of instrument as well. So Folin developed a method for analyzing blood sugar using a ferrocyanide solution. It turned blue and then was compared with a sample in the colorimeter. Now, it's interesting to note that this particular blue has its own history. So it was one of the first stable blue pigments that was discovered, and it was used since the 18th century. It's got its name Prussian blue because it was used as a dye in the uniforms of the Prussian army. And this brilliant blue was also used in many works of art. So after 1928, it provided the means to measure sugar in blood. Now, a good colorimeter was pretty expensive, and labs usually also had to make their own standard solutions for the comparison. So they also required skill to use, and comparisons took time. So this was a method that required expertise and facilities, almost mainly for the clinical lab. An alternative was to use sets of permanent standards, like this one from the company Lamotte in 1929. So this kit was developed in direct response to Follin's methods with Prussian blue, and each of these vials contains a different concentration of a ferrocyanide solution. So this test kit comes with all of the equipment and reagents necessary to do the Follin blood sugar analysis. And then the user compares the color of his sample with the standards to get the sugar percentage within 0.5%. This was also a professional kit because blood sugar analysis was tricky and the equipment was comparatively expensive. So most individual sugar monitoring was still done through urinalysis. But the problem of standardization was there as well. This quantification was done through color comparison charts. Now, I'm going to show a few slides now where I'm going to quickly go through a few of these comparison charts so that some of the issues become apparent and you also see the sheer variety of scales and models that were out there. So one physician who was particularly interested in developing methods that could be used by patients was Michael Somogyi. Somogyi wanted his patients to test their urines multiple times a day, up to 12 times a day, and to keep very, very tight control of their sugar levels and diet. In the 1950s, he developed a very simple and inexpensive urinalysis method based on caramelizing the urine in an alkaline solution. And these are 3 comparators he developed for his method. There's a permanent one that has vials of an iodine solution in a wooden base, a paper chart, and then a specially designed comparator where the user dripped urine into the white indentations and compared its color to those in the numbered shaded indentations. And this was made of Bakelite, so it could be easily cleaned. Here are some of the color charts that I've shown already in this talk. And one problem with all of these paper-based standards is that they deteriorated over time and the colors changed. Another problem was how to get the color right in the first place, because these are mass-produced printed documents. So to solve this problem, Helen Frey, for example, turned to a standardized color chit book that had been made especially for product designers in the very colorful 1940s. This is a different approach to colorimetry, but much like the liquid color standards, these little chits had numbers. And by using the same color book, biochemist and printer could communicate about the exact shades on the test kit color chart. So Free used a version of this manual to communicate with the printer who made the color charts for the dip and read test strips. Now, I want to return now to the question of shifting skills. So changes in the mode of conducting sugar tests had shifted much of the skill and expertise from the person doing the test to the chemist in the pharmaceutical laboratories who are making the test. And this allowed a new group of users, diabetic individuals, to assume responsibility for day-to-day testing. To do this, they needed to acquire the skills of color reading in all of these different kinds of color charts. But color perception depends on a number of factors. So actual colors do not always align with the discrete steps in the color charts. Bad lighting, fading, diabetic retinopathy, and wishful thinking can alter perceptions. So visual color perception is always individual, and medical professionals were rarely satisfied that patients were being consistent and accurate in their interpretations. And this is a huge problem if accurate tests are needed to determine the right dose of insulin and to avoid diabetes complications. So this is where a new group of experts enter the picture. This is physicists. In the mid-decades of the 20th century, and especially around and after the Second World War, physicists engaged in optical research for the industry and for the military, and they developed various instruments capable of quantifying light waves, among them photoelectric colorimeters that could measure color and quantify it and express it as numerical standards. So color was essentially reduced to light waves, and the measurement of light was standardized. And this had a tremendous effect on medical lab work. So the basis was still in biochemistry, but photoelectric optical instruments now did the interpretations. So clinical chemistry was flooded with electronic colorimeters, spectrophotometers, and other instruments that measured color changes according to programmed-in standards, and they could do that in large batches. So these developments in optics were the basis for the first photoelectric reflectance meters for blood glucose measurements. This is the first one, and it was developed by Tom Clements, who worked in instrument research and development at Ames Labs. So one of the motivating factors behind this instrument was solving a problem that had emerged with another Ames product. In 1965, Ames chemists developed dextrosticks, which was a dip and read strip for blood glucose. And dextrosticks results were much more difficult to interpret than the results of the urine test strips. For that reason, they were used primarily by physicians, but blood sugar measurements were preferable to urine sugar measurements because they're more accurate and immediate. And for some patients, this immediacy was needed to achieve adequate control of sugar levels. So these first electronic blood glucose monitors were developed for hospital use. They used the same dextrosticks test strips that could be visually read, but the strip was put into the monitor and read off by a photoelectric cell instead of the human eye. Then the results were communicated through the deflection of a needle. Later models then introduced digital display. So these early meters, the first generation of meters, were very expensive, and they were not something that an average person with diabetes would be able to use just due to the cost alone. By the early 1980s, however, glucose monitors designed specifically for patients and home use came onto the market. So these are some of the earliest home models. They are pretty large because they contain an actual optical photometer. So by the 1990s, biosensors allowed monitors to become much smaller and more flexible design, and they led to the great variety in design that we see on the market today. But I'm going to return to the theme of this talk of shifting expertise and shifting responsibilities. What we see here is that the results interpretation is now being standardized and put into an instrument. It's no longer dependent on the skills and expertise of an individual user. And this is frequently interpreted as a sort of de-skilling and a simplification, but that's not entirely accurate. So the monitor itself embodies a lot of physics knowledge, as it also embodies a lot of chemistry knowledge. But the end user now just needed to learn to do other things. So compared to the dip and read test strips, which were extremely simple, using these monitors requires a lot of routine skills. And this is what a user now needed to learn. Sterilization, calibration, machine maintenance, a lot of tasks centered around making sure that this apparatus continued to work accurately. So to sum up the development of sugar monitoring over the course of the 19th century, we see it moving from being based in expert biochemistry, so where it was largely undertaken only by clinical chemists for diagnostic purposes and by a diabetes specialist for occasional measurements. With the discovery of insulin and the possibility of intervention and control of diabetes, regular monitoring became crucial for the therapy to be effective. This biochemical expertise then became increasingly embedded in the tests themselves, and the tester increasingly only needed to unleash it and then interpret the results. This was crucial for the shift of monitoring responsibility from expert physicians to patients as experts. So with blood glucose monitoring, problems of results interpretation became more pressing since it was more difficult. But then the expertise with these interpretations became embedded in photoelectric monitors. End users then had to learn to run and maintain these little machines. So I'm a historian, and I'm stopping here in the 1980s, but these trends can be followed on to the present day. So each innovation in monitoring shifts certain expertise and responsibility from one place to another, while new tasks and new skills and responsibilities turn up in a different part of the process. And thank you for listening.
Thank much, Professor Neswell. Very interesting. And what I want to do now, we'll move into a discussion period. But first, I just want to remind people there's a way to submit questions through a QR code on your, you can use your phone to get to. The QR code is there on the screen. Or you can just use your phone on that. Or if you can just go to www.sli.do and hashtag RSC2. So that's one way to submit questions. So, and also before we get into the question, I just wanted to, I got cut off earlier on. I had a sort of errant phone call come into the house here and interrupted my introduction of Dr. Lewis. Most of you probably know something about him already, but I just want to remind you a bit more about his background. He's currently the head of the Departments of Medicine, the director of the Banting and Best Diabetes Center, ET. Became that in 2011. He's full professor in the Departments of Medicine and Physiology, University of Toronto, and holds the Sun Life Financial Chair in Diabetes and the Drucker Family Chair Diabetes Research. He's been involved with a number of important discoveries, so to look that up, I don't want to spend too much time going through that at the moment. I have limited time to go into a discussion. So anyway, thank you very much for your papers. They were very interesting. As I mentioned earlier on, I'm involved with the Defining Moments Canada Insulin 100 project, and so I've kind of been immersed in all this. So I'm trying to maintain my moderator position here and not get too caught up in what I know about this. So I just want to focus on what we've talked about here. I have a couple of questions kind of for each of the presenters here. Just a bit provocative perhaps to get things going. Start with Gary. Mentioned in your presentation about basically the basic idea of Banting was a bit erroneous in the sense of didn't have to be ligated, and you have to have it degenerate to proceed. But does that really matter in the sense of to the whole process of the story? I mean, the point was certainly that it got the process going, that he had an idea that was intriguing enough to present to MacLeod, and then see what happened after that. So I don't know, what are your thoughts about that?
Thanks, Chris. Hopefully you can hear me. So the question is, the fact that Banting's hypothesis or original idea proved not to be correct, was that a problem? And as I try to indicate, I don't think it was at all. What's important is to have a testable hypothesis. So it was a testable hypothesis. And I can't tell you, at least in my own lab, how many times our hypothesis is wrong and how many times we've got opposite findings that have turned out to be way more interesting. So a hypothesis does not need to be correct. It's simply a hypothesis, which is an idea, and it has to be testable. It does not have to be correct. And the important point here is it got Banting very enthusiastic. He was a very enthusiastic fellow, and it got him going, I mean, amazing sacrifices he made financially. He moved towns. He put a lot into this. By the way, he didn't really see it as a testable hypothesis. As I said, he simply wanted to cure diabetes and to isolate an internal secretion. So he behaved much less like a scientist and much more like a driven man to achieve something. The answer is no. I don't think it made a difference.
Exactly. Well, I sort of feel the same way. And yeah, Banting indeed was very driven, very unique person. Actually, before the session, I was down at the official library doing interviews for a new documentary about Banting's life. So I'm, as I said, I've been immersed in a lot of this already. So, okay, I'll just have a question just for Elizabeth to kind of start things to get you to think about something. As diabetics manage their use of insulin, what were some of the practical and pragmatic and economic challenges that were involved in, especially in the early part of the story here, in selecting and using and paying for evolving urine and blood testing kits and technologies? Like, I mean, you talked a lot about the technologies and how that changed, but what was sort of the perspective? Have you looked at the perspective from the diabetic themselves and how the challenges, you know, how do you do urine tests like 14 times a day, stuff like that? I mean, I can just imagine, for our child, perhaps, trying to do this. What kind of thoughts do you have about the sort of the practical side of things?
Various things have turned up at various stages. I know for Sumoji, he was very interested in keeping the cost of the test low so that the patients would test often, but also that the patients were absolutely sick of constantly doing these tests. For Sumoji, I read some documents from about a year or couple of years of one of his patients who was writing to him regularly. And this was very, very burdensome, the number of tests. So every so often he would negotiate down to only doing 8 tests a day or 6, down from the 14. So yes, that became very burdensome. This was with the, so Sumoji had his cheap method that he developed precisely for the costs. Another thing that turned up with the strips. Now the strips are very easy, but this also adds up in cost over time. So patients would split their strips. And I also spoke to someone quite recently who was diagnosed maybe in the 80s where there's still strips, who said, yes, he cut them down to like a quarter of the size. So he'd divide each strip into 4 pieces to try and save, and assume that he had developed the expertise to interpret from something that small. There were devices developed for patients to split their strips, some in the Sears catalog, like a strip splitter, and quite a lot of text written by physicians saying, don't do this, this is dangerous. So cost was always a problem there. I mean, it's still a problem with the test strips. There's a gray market for test strips in the United States where diabetics have to cover a lot of their own costs. So, and each glucose monitor is, they only take certain strips. So if you use one monitor and then you change and you have the strips, you have to get rid of them. So these are sold on the market trying to reduce costs. In regard to the devices, the blood glucose monitors, the first ones were extremely expensive. So those were not sold. Even into the 80s, the glucometers and the AccuChecks were quite expensive, but insurance companies were quickly convinced in the US to cover the costs for very severe diabetic patients. So they took over the money, and that was the way that the patients were able to afford it.
And was there any kind of standards established for the test kits? I mean, you pointed to some from Lilly and the strips themselves, but was there a kind of a standard or a standard established or trying to establish a standard between companies doing the same kind of tests? So you're not sort of beholden to a particular type of test from a particular company. And if,
no, they were
transferable. Is it standard that goes around that you just have different sort of slight variations perhaps on it, or?
They were proprietarial.
Yeah.
So no, there is one case of cooperation, and that was with the clinistics and the test tape. So the first dip and read models, one from Eli Lilly and the other from Ames Miles, that because the techniques, because it was around the same time, they agreed to share licensing. But in most cases, there were fine differences in the indicators that were used so that the color charts did not align. And this was also a problem, that a patient who learned to use one particular brand and their color scale might switch and misinterpret the color scale. Because if you look closely, you wouldn't have had the opportunity, but if you look closely at the Clinistics color chart and the test tape color chart, the colors look the same, but they actually go in the opposite direction. So what's low in one is high in the other. So this was a big problem for patients. And when they learned to read one, they were disinclined to move to another because they had to relearn how to interpret the results again.
It seems complicated to manage.
Yeah.
Okay, there's this one. A question came in through the app here for Elizabeth. Actually, well, I've got you here. Might as well ask you this one, Elizabeth. Given the frequent changes in the testing devices and protocols over the years, how did physicians feel about glucose testing, and did they struggle with problems due to the adoption of these new technologies?
Okay, how I don't know whether physicians struggled. I don't know if they would have admitted it publicly. There were quite a few diabetes specialists and also diabetes nurses in the 20th century. And they did look very closely at all innovations, and they would be thoroughly tested, compared to see which one's best, which one works best for this kind of patient, which one can patients be trusted best to use without mistakes. So I found with people involved in the actual diabetes care with the tests, they really appreciated the innovations. They were not sure how the patients would, and the patients themselves were often very reluctant to move from one to another because it required learning a new skill set. Once they felt secure with it, with one, they did not want to take a chance with another. And I've heard anecdotally that this is also still the case, that some patients don't want to move to certain to constant monitoring because they are very comfortable with their current glucose monitors and know how to interpret everything and don't want to take the risk. So this has been a constant. It's more the patients than the medical practitioners who are assessing on the go whether or not they want to change.
Yeah, it just seems that change is always a challenge with the whole insulin and diabetes management process. When things are improving, and that's all great, but then it throws people off. They've been used to doing something for a long time, right? So
yeah, there's a very good text out. There's a couple of texts out about currently the self-monitoring and the constant monitoring, and just questions about for patients with whether or not they want to change or whether they feel that they can interpret the results and the state of their body and what they've been doing comfortably with the method they have now. So there is a reluctance in among some patients and some practitioners to change from what they know.
Yeah. Okay. Back to Dr. Lewis. Kind of a provocative question again for you, just to what you think. I'm just interested in kind of the key elements that enable banking's idea to develop. And so what was I think the weakest link in the chain of events that have broken, sort of that led from the idea through to the first use in Leonard Thompson, through to the actually having a supply of insulin available? What was kind of the weakest link in this chain, you think, that could have broken or severely or compromised the whole Toronto story? Because it just seems to me there's very fragile points along the way. And just from your perspective, what kind of where would you think that might have happened?
It's a good question, and I'll answer it, but I do just want to take a minute to say that our moderator, Chris Ruddy, is because you weren't introduced, is a foremost expert, and Professor Ruddy is a foremost historian in the discovery of insulin, having trained with the late and great Michael Bliss. And so I'm not a historian. My colleague and I, Pat Brubaker, have tried to interpret the events, give our little spin on it. But if you have questions, and Chris has a fantastically rich website. So I do want to say that. But in answer to your question, and I'm glad I didn't forget it while saying that, is the weakest link, in my opinion, would have been if any of those four didn't exist or didn't participate. So each one on their own would have been insufficient. Okay, and you can talk about each one, right? So without Banting's sort of drive and laser focus, it wouldn't have got started. And I mean, I didn't tell you, but the lack of success, the number of dogs that died fast was incredible. I mean, you had to be really tough to do those experiments. And I know that Charlie Best really supported him. There's wonderful stories, and he relied on Charlie a lot, and Best played a major role in the production. They couldn't have done it without the resources of MacLeod and the expertise and the guidance. And, they didn't know what they were doing. And MacLeod was great. And then Connaught, of course. But Connaught couldn't have done his experiments with alcohol extraction without the others. So the weakest link would, if any of them didn't come together, they wouldn't have made the progress. So it was the team. It's teamwork. And That's my opinion, but may not be historically accurate.
Well, as you're referring to Michael Bliss, his term, the glory enough for all, is kind of the term that applies to the four of them. And you can expand that out to people like the Fitzgerald at Connaught, and there's a whole range of people that were vital to that chain, moving along from the lab to Lilly and to Connaught, and the role Connaught played overall in supplying insulin all the way to Canada from the very beginning. So there's a lot there, and there's a lot more on the Defining Moments website, that, as I mentioned before. So it's definingmomentscanada.ca. So feel free to explore that. There's lots of content going up on to explore this story further. All right, so we're almost out of time here. I just want to say one thing briefly, as I've been asked to say. Thank you very much, Dr. Lewis and Professor Neswald, for our presentations today. And I just wanted to say that at the end, there's a 15-minute break coming up in this webinar. So please choose from the next 3 streams. There's more streams that'll be coming up, available when we come back in about 15 minutes. So thank you again, everybody, for joining us, and we'll carry on. Okay.
Thank you.
Take care.
Bye-bye.
Bye-bye. Thank you.